Hello everyone, this is the third part in a sequence of videos that I'm doing on cellular respiration. I start by looking at aerobic respiration, then in the final video to this sequence I'll look at anaerobic. But what we're going to do in this video is think about the final stage in aerobic respiration, and that's known as oxidative phosphorylation. Oxidative because along the way we're getting a series of oxidation reactions, so things are losing hydrogens, losing electrons. And phosphorylation because we are adding phosphate to certain molecules. Now, ultimately, the aim of this process is to produce ATP, and it's ATP that is going to fuel biological processes. If we think back to my first video on this in part 1. So for all the metabolic processes that we need to do, we need ATP, and that's really the purpose of this. Now, if you think of the general, very simple equation for respiration, like glucose and oxygen give CO2, water and energy, that you learn about lower down in the school. This is really where the oxygen comes in, because oxygen is going to serve as a final electron acceptor in what's known as the Electron Transport Chain. But we'll come to that in this video. So what we're going to do is build up the sequence of events, starting with this in the middle of the screen. What I've got here is a phospholipid bilayer. So I've got a portion of a cell membrane. Now what I need to do really is orientate ourselves on the screen. So you'll have just seen that I've flashed up the names of certain sections. So at the top, what we've got is the matrix of the mitochondria. This membrane is a portion, it's the actual inner membrane of the cristae of the mitochondria. At the bottom, we've got the intermembrane space. So I'm going to quickly draw the mitochondria to show you these three sections, just to make that a little bit clearer. So here we have a picture that I've just inserted there, a little sketch that I've drawn previously of a mitochondria. Not graphically the best, but basically we've got the blue matrix, or what I've coloured blue in the middle is the matrix, and you can see the foldings of the black membranes, the cristae. So where I've numbered number 1 on that little diagram in the area of blue, that's representing the top layer of this whole screen, if you like, the matrix. I've drawn a red number 2 on the black line. That's representing this central picture that I have of the membrane. And you can see the yellow circles of this membrane represent the phospholipids, and the 5 big coloured blobs that you see in the middle essentially represent protein structures, intrinsic proteins within that membrane. And then where I've put a red number 3 on the diagram on the top of the mitochondria, that's a sort of space between 2 folds. And that is the bottom layer of this whole screen. That's what's known as the intermembrane space, which I've just dotted and coloured green. So if we were to give themselves numbers, this is number 1 on that picture of the mitochondria, this is number 2, and this region here is 3. So that's where how we're orientating ourselves, and I'll leave that picture of the mitochondria up there. Now this picture is going to get quite full, but we'll build it up step by step. So what I said was that through Krebs we produce a good amount of reduced NAD and FADH2. So we need those coenzymes, because what we're going to do now is oxidize them to produce energy. So we have in the matrix this NADH, and that NADH now will become NAD+, and equally FADH2 will now become FAD. Both of these reduced coenzymes become oxidized. Now when they become oxidized, what they've essentially done is lose hydrogen atoms, and those hydrogen atoms split into protons, which are H+ ions essentially, because we can see that the NADH, when it's become NAD+, loses H, and the same with FADH2. When it becomes FAD, we've lost H. So we split it into protons, but we also get some electrons, which are E-. or has a symbol of E minus. And those electrons move what's called through the Electron Transport Chain. And it's made, that Electron Transport Chain is made up of various protein carriers. And that's what this blue, pink, orange, green, that's what these circles represent. Now technically there are actually only 3 electron carriers in the sequence, but I've put 5 just for a visual purpose because the last 2 are where we get something special happening, which we'll talk about. So these electrons get picked up by this carrier and they move along this transport chain from carrier to carrier. And as those electrons move along the transport chain, they lose energy. And that energy is used to pump those H+ ions from the matrix through the inner membrane of the crystal and into the intermembrane space. So I won't draw all of them because it might get a little bit too messy on the diagram. I'll just draw these 3. But essentially those H+ ions get pumped into this intermembrane space. So you can see we're building up this concentration of hydrogen here on the bottom of the screen. So we've oxidized NADH and FADH2. We've provided some protons, which are the H+ ions, and electrons. And that passage of electrons from complex to complex causes those hydrogen ions to be pumped into the intermembrane space. Now, one term that you should know for the exam is chemiosmosis. That is the name we give to the process of ATP synthesis, which is what we're doing here. We're trying to produce ATP ultimately via electron transport and the use of concentration gradients. So making ATP using these electron carriers with electrons jumping from one to another, that process is known as chemiosmosis. And I think I'll just make a little note at the top underneath the picture of the mitochondria here. So that term that I've just referred to is chemiosmosis. So the name given to the process of ATP synthesis via electron transport and the use of concentration gradients, which we'll talk more about in a moment. So now we've got this supply of H+ in the intermembrane space. What we have now is a concentration of protons higher in the intermembrane space than actually in the mitochondrial matrix itself, because that's where they started. So now we have lots of hydrogen at the bottom and less at the top on this picture. And it's that allows us to form what's called an electrochemical gradient. So I'll write this down here. So because those hydrogens have been pumped down here, we get what's called an electrochemical gradient. Now, before we deal with those H+ ions, it's important to note what happens to those electrons in the electron transport chain. So these electrons, now, if we move them to this green one here, these electrons now combine with oxygen. So we get something very special at this stage here. Oxygen will combine with 2 protons and those 2 electrons, and it will form water. So I'll write at the bottom the equation that we've got, and I'll write it in green just for colour's sake. So at that point there, what we have is half O2, so oxygen combines with 2 H+, 2 protons, plus the 2 E minus, 2 electrons, and what that forms is H2O. So this is the point there where we produce water. So in the mitochondrial matrix, at the end of the transport chain, protons, electrons, and oxygen from the blood combine to form water. So oxygen is said to be the final electron acceptor. And that's, I think, something we should really make a note of, that O2 is the final electron acceptor. So we've had electrons move from carrier to carrier. As the electrons have moved, they've lost energy, and that energy has been used to pump H plus ions into the intermembrane space. Those electrons are then picked up by oxygen to form water, so oxygen is the final electron acceptor. And now in the final stage, we can deal with that electrochemical gradient of hydrogens. Now this is what happens. This last block on this picture, this big blue circle that I have at the bottom, or have on the side rather, that there represents ATP synthase. That's what that blue block represents. Now, ATP synthase is this protein, ultimately, this enzyme that's going to be used to create ATP. We have a large concentration of H plus ions in the intermembrane space. Now, because we've got a high concentration here and low concentration in the matrix, that hydrogen is pumped back into the matrix along the concentration gradient. So this H plus now gets pumped through here into, I just happen to be hitting the picture of the mitochondria, but it's not going to that specific point. Although actually I seem to be technically hitting the matrix there, but the H plus, or those hydrogen ions, are pumped back into the matrix along a concentration gradient through ATP synthase. Now that releases energy, allowing ATP synthase to activate, because ATP synthase is an enzyme, and it activates to phosphorylate ADP. So basically adds an inorganic phosphate to ADP to make ATP. So any inorganic phosphates can add to adenosine diphosphate to form adenosine triphosphate. So ultimately what we're getting on this last stage is ADP and P forming ATP, which is what we want. It's the purpose of this process. So all those hydrogens from the intermembrane space get pumped back right up through the inner membrane of the crystal, back into the matrix, activating ATP synthase, producing lots of ATP. And in fact, it should be said that about, if we were to put numbers to it, for each molecule of glucose that you start with in respiration, 28 molecules of ATP are produced by this process alone, oxidative phosphorylation. So that's ATP made from reduced NAD or NADH and FADH2. So in this process, what we're actually producing is 28 ATP. So if we add that to the 4 we made previously through glycolysis, link reaction and Krebs, that makes a grand total of 32 ATP. An interesting sort of side, the number of ATP produced per reduced NAD or reduced FAD was thought to originally be 3 and 2 respectively, but new research has shown that the figures are actually nearer to 2.5 and 1.5. But basically we make in total, by this point in time in respiration, 32 ATP. And that's key to this. Now, one question examiners like to ask is, what would happen in terms of the electron transport chain if oxygen supply suddenly diminished? So now we're thinking about maybe leading into anaerobic respiration. Well, let's think this through. If there was no oxygen, there would be no terminal electron acceptor. So there would be nothing for the electrons to be taken up by, if you like. So the electrons would remain on these protein channels. Now that's unstable. The cell wouldn't allow that. We can't have electrons in this membrane in this state, if you like, because then these channels would be permanently reduced. If the electrons then are just stuck and aren't moving, the hydrogen ions aren't then being pumped from the matrix into the intermembrane space. And if we don't have that, then there's nothing to pump back through ATP synthase. So ultimately, if we're stopping those hydrogens moving because the electrons are stuck, we're not producing any ATP. So essentially we're shutting down this whole phase. And actually, because we're shutting down this phase, we would also shut down the link reaction, because the link reaction too happens in the matrix of the mitochondria. And what we'd do in that case is get this build-up of NADH and FADH2, a build-up of these reduced coenzymes. Now, if you think back to part 1 in the, on this sequence of videos on respiration, I said that you needed NAD for glycolysis to happen, because you need NAD to convert to NADH, or glycolysis would shut down. And that's going to lead in perfectly to what happens during anaerobic respiration, because we can't clearly shut down the entire process, or living organisms would die. We need energy from this process. So in the final part of this sequence of videos, I'm going to look at what happens in anaerobic respiration, so without oxygen. But this video ultimately is showing you part 3, oxidative phosphorylation, the production of ATP, 28 ATP, in this process using reduced NAD and reduced FAD through what's called an electron transport chain. Okay, hope all that helps.